I'm very pleased to introduce our next speaker, Dr. Stefani Spranger. She's a professor in Department of Biology at MIT, and her lab focuses on tumor immunology, dendritic cells, and the interaction between tumor cells and immune cells. And Forrest has been instrumental in hijacking her and making sure that now she becomes excited in working on GBM. This is our goal towards Stefani. Please, Stefani.
Thank you, Antonio, for this great introduction. And it's really a pleasure being here at MIT. And Antonio's right. Forrest got me excited, and then I got Amy, a trainee in the lab, excited to work on GBM. So we're now shifting towards this very, very horrible disease. I will, for disclaimer, mostly talk about non-GBM related work and mostly talking about immunology. So I will be going slow because I know cell-cell interactions of the immune system can be daunting. So we in the lab think very heavily about anti-tumor immune responses along the cancer immunity cycle. And I want to introduce on how we think about this. We think initially cancer cells, dying cancer cells, cancer cell debris has to be sensed by innate immune cells, specifically a very specific subset of innate immune cells called cross-presenting dendritic cells. And these cross-presenting dendritic cells infiltrate the tumor mass. They sense dying cells. They pick up tumor cell debris, and that induces an activated and migratory profile in those DCs that then allows those dendritic cells to go to the closest secondary lymphoid organ. In most cases, this is the tumor-draining lymph node. In the tumor-draining lymph node, T cells that see the cognate peptide MHC complexes that are now cross-presented on the surface of the dendritic cells with peptides that are derived from the tumor are activated, they expand, and they differentiate along certain very preformed differentiation trajectories. Once activated and fully expanded, those CD8 T cells then home back into the tumor microenvironment. And in many cases, the homing signals of those effector T cells to go back into the tumor are also dependent on dendritic cells and sometimes myeloid cells that sit within the tumor microenvironment. And then once they're in the tumor, these CD8 T cells have the capability to recognize antigens on the cancer cells and eliminate cancer cells. And this is where the power of immunotherapy and the immune system recognizing cancer really comes from. If this would work, as I just described it, cancer wouldn't be a clinical problem because our immune system could get rid of the cancer. So one often therapeutically targeted example of how the immune system fails to eliminate cancer is T cell exhaustion. This is not, as I depicted it here on the slide, a one-time switch in the tumor microenvironment. This is actually an entire trajectory that happens as the T cells are being activated. But once they reach terminal T cell exhaustion, the T cells are long-lived. They stay in the tumor, but they can no longer kill the tumor cells. And that's where most current immunotherapies across many, many different cancer types are actually acting on to prevent this terminal T cell exhaustion from occurring. The first cancer type that I want to talk about is non-small cell lung cancer, which, if you take a 3,000-foot view, is actually an immunotherapy-sensitive cancer type. However, if you look really closely, only in about 30 to 40% of patients is dual checkpoint blockade immunotherapy consisting of an anti-PD-1 and an anti-CTLA-4 antibody actually efficacious in the long term, which is certainly not enough. And we as a group wanted to understand how can we get this further up, and this is just another way to illustrate this. And fairly early in our studies, we wanted to compare this across different cancer types. Because our hypothesis was, is there maybe something about how the immune system is engineered for each specific cancer and each specific tissue site that prevents responses towards immunotherapy? So Maria Ceruglia, a previous student, went through many clinical data sets where cancers were treated with dual checkpoint blockade immunotherapy, so similar to what I showed you on the previous slide. And here just reported the objective response rate following this dual checkpoint blockade immunotherapy. And what struck us was the two most responsive cancer types were Merkel cell carcinoma and cutaneous melanoma. Merkel cell carcinoma, much less known, is a skin cancer. Cutaneous melanoma is a skin cancer. And then everything else sort of dribbles off, with the high mutational burden tumors obviously rising somewhat more to the top. And non-small cell lung cancer actually falls somewhere in the middle of the responsive cancer types. And that really solidified our hypothesis that maybe there's something about the specific tissue architecture and the immune system for each tissue that mediates response or non-response. And in addition, if we zoom in clinically into what is known about the immune archetypes that are within non-small cell lung cancer patients, we can stratify them into 4 different discrete populations, with this one here being the best understood, where we have T cells, we have exhausted T cells, and we have, and this is important, upregulation of PD-L1 on the tumor cells. And PD-L1 is a downstream target of interferon gamma. And interferon gamma is made by CD8 T cells that are properly activated. And that, if you put these pieces together, means there are productive T cell responses in those tumors if we have PD-L1 upregulation. The other cancer type that is far less understood but clinically reported is termed non-functional T cell response. And these cancer patients have, non-small cell lung cancer patients, have T cells in their tumor mass, but they lack the upregulation of PD-L1. And this was what we thought maybe the tissue-specific immune constraints might play into, because why are these T cells not properly activated? Why are they not doing what they're supposed to do? So first Brendan Horton and then later Maria Zaruglia worked on this problem. And the way Brendan approached this is he used a cell line that is derived from an autochthonous tumor model driven by KRAS and P53. And then simply implanted those cells orthotopically. We can do this by tail vein injection or intratracheal injection. We get the same phenotypes. Or subcutaneously, which is an absolutely artificial site, but it models the immune response towards a cutaneous tumor. And then treated with dual checkpoint blockade immunotherapy, similar to the patient data that I showed you. And we were quite surprised that the lung tumors were absolutely resistant to checkpoint blockade immunotherapy, while the flank tumors reduced in size by about 50%. And this was even more stark when we looked at actually what happens in the tumors themselves, because we found quite a lot of T cells in the lung tumor lesions, but they failed to expand following checkpoint blockade immunotherapy, versus in the flank tumors, we had fairly low at baseline, but they drastically expanded following checkpoint. And that gave us the indication this is probably a T cell intrinsic defect to respond to therapy, and not something the tumor imposes on the T cells. So we then took an unbiased approach to understand how are these two T cell responses different. We used single-cell RNA sequencing in collaboration with Chris Love's lab. And these two clusters down here are the activated CD8 T cells. We then stratified them back on which tumor site that they come from. Cluster 1 is mostly from flank tumors, while the lung tumors mostly had cluster 2 T cells. And then, of course, this is a polyclonal T cell response, so we then engineered in a model antigen, which allowed us to really go after the tumor-reactive T cells, and we saw no differences. And then when we looked at what are the differentially expressed transcripts, we found that the cluster 1 T cells have all the markers that we would canonically associate with an exhausted T cell response. And in contrast, and we're still trying to piece this all together, the cluster 2 T cells have some markers of tissue-resident memory T cells, or central memory T cells, or tolerized T cells. But they're not effector T cells, and they're not exhausted T cells. Is this relevant for humans? We can generate signatures based on these differentially expressed genes, and then impose them onto non-small cell lung cancer patient data sets, and actually see that in the large majority of the T cells, the TIL that we find in the non-small cell lung cancer patients themselves have this dysfunctional phenotype, as we called it, while only a small fraction of the T cells have this truly exhausted T cell response. And that indicates to us, even if the patients have a mixed population, we might not be leveraging all the T cells that are in a non-small cell lung tumor patient with current checkpoint blockade immunotherapies. Now, The lung is a very different site than the flank for tumors to grow. So we wanted to understand when in the T cell activation cascade does this difference occur. And for that, we used a trick. We used, again, this model antigen called SIY. And we used T cells that are transgenic for a T cell receptor that is specific for this one peptide. We labeled those T cells with a proliferation dye and then transferred them in and let them be activated for 3 days in mice that have tumors. And then look specifically in the tumor-draining lymph node. And what we found is that the activation profile here read out by dye dilution, because every time the cell divides, the dye gets diluted. We see roughly similar kinetics of T cell activation over time. But then when we pull out the activated T cells from those lymph nodes and do sequencing on those T cells, and I'm still blown away by the number of differentially expressed genes that we found, just 3 days, 72 hours after we put them in, we found over 1,000 differentially expressed genes, indicating that this bifurcation of taking an exhausted T cell differentiation versus a dysfunctional tolerized T cell activation happens very rapidly as the T cells are being activated and probably within the first 3 to 4 cell divisions of T cell activation. Two of the very relevant functional markers that we used for subsequent studies are CD25, which is the high affinity IL-2 receptor, as well as granzyme B, which is a cytotoxic molecule. And these are higher upregulated in the inguinal lymph node when the T cells become activated. Maria then really wanted to understand those dynamics in the lymph node. Like, what imposes this tolerance-promoting microenvironment that pushes the T cells down this absolutely non-effective differentiation path. So to first establish, she identified that cross-presenting dendritic cells indeed are the cells that drive T cell activation in both lymph nodes. And then she looked at the activation signals that these DCs provide to the T cells. The first thing we looked at was just antigen density and antigen availability. And for that, we used a trick where we engineered the tumor cells to have ZS green, which is a pH-stable fluorophore, so we can track it in the dendritic cells. And when we look then in the dendritic cells in the corresponding draining lymph nodes, we find that the mediastinal lymph node has more antigen. We looked at various different ways of how you can sparse this out. We always found more antigen, so we excluded signal 1 as the main problem. But then when we looked at signal 2 and 3, meaning co-stimulation as well as cytokine profiling, we identified that there's far less co-stimulatory signals that push T cells into an effector state in the mediastinal lymph node, and far less IL-12, which is a key cytokine associated with cytotoxicity in CD8 T cells. So that by itself led us to hypothesize, well, maybe the dendritic cells are intrinsically different between the two lymph nodes, and Maria has done a lot of experiments disproving this hypothesis. And then we were looking for a third cell type in this interaction that would actually or could actually impose this immunosuppressive feature on the dendritic cells. And we ended up hypothesizing that potentially regulatory T cells could suppress dendritic cell activation very specifically. And to first test that, Maria established an in vitro co-culture assay where we sort out dendritic cells as well as regulatory T cells from the same lymph node. And we specifically used dendritic cells that have taken up antigen, and then co-culture them with tumor-reactive T cells, either in the presence or absence of regulatory T cells. Here on the left, you see that indeed dendritic cells intrinsically are not dysfunctional. If we only have DCs and CD8 T cells, we get really nice effector T cell differentiation. If we add regulatory T cells back in, and this is a very, very low ratio of regulatory T cells, we get very significant suppression of effector function, but not proliferation. And this was exactly what we saw in vivo in our mouse model. We did a lot of other in vivo experiments that I'm not going to show for time constraints, but to summarize, we have established that the cross-presenting dendritic cells that sit in the tumor-draining lymph node interact with both regulatory T cells as well as the CD8 T cells. And the regulatory T cells, via an MHC class II dependent interaction, inhibit the upregulation of CD80 and 86, as well as the production of IL-12, and by doing so, suppress the CD8 T cell activation. So why can regulatory T cells in the mediastinal lymph node do this better compared to Tregs in the inguinal lymph node? That was our next question. So we then thought, well, maybe it's density of regulatory T cells or number of regulatory T cells. Neither of these was the right answer because both of these are about the same between the two lymph nodes. And then we again collaborated with Chris Love's lab and did single-cell RNA sequencing paired with TCR sequencing. And that was really key because we were able to understand by clonotype analysis which population of the Tregs is actually expanding in response to cancer. And quite nicely, we found that it's the same population of Tregs that are responding to the cancer. They even have shared clonotypes, meaning they're seeing the same antigen. But when we compared exactly this population down here for their differentially expressed genes, we found the Tregs that sit in the mediastinal lymph node are more skewed towards the Th1 phenotype, allowing them to be potentially more suppressive for CD8 T cells and cross-presenting dendritic cells. I'm going to show you the flow validation for this. We find more CXCR3, which is a key Th1 marker, as well as TBAT on those regulatory T cells in the mediastinal lymph node. Th1 is downstream of interferon gamma. So we then hypothesized, is there a higher level of interferon gamma in the mediastinal lymph node? Maria established an ELISA to actually assess this, and indeed there was more interferon gamma present in the mediastinal lymph node. We then said, well, there's more natural killer cells responding to the cancer that we put in. Maybe that's a consequence of the tumor growing in the lung. We looked in naive animals and actually identified the difference was bigger in the naive lymph nodes, indicating this is a true tissue-specific constraint or manipulation of the immune system. And because the lung is a mucosal-associated lymphoid or mucosal-associated tissue, we hypothesized this might be a consequence of having commensal bacteria somewhere in the animal. So we looked in germ-free animals, and indeed we identified that having no commensals reduces the levels of interferon gamma in the mediastinal lymph node specifically. And this is what we're currently following up on. So to summarize this part, what we identified here is that the regulatory T cells suppress DCs very specifically, and by doing so inhibit the dominant CD8 T cell responses in this setting. But how can we therapeutically fix this problem? And this is something where collaborations with engineers is really an amazing tool to get at those mechanistic insights. So we found in our differentially expressed gene list that cytokine receptors like CD25, so the high affinity IL-2 receptor, and IL-12 were differentially regulated. So we collaborated first with Dane Wittrup and later with Forrest on this to understand, can we use half-life extended cytokines to actually push these T cells into a more effector-like phenotype? And indeed, we can upregulate CD25, we can upregulate granzyme. And this very nicely translates into a very significant survival benefit when we combine these two cytokines together. And then Alicia in Forrest's lab really worked with Brendan on this to understand it's not just 2 + 2 is 4, it's actually 2 + 2 is 10. Because IL-2 by itself changes the signaling in the T cells a little bit. IL-12 changes the signaling in the T cells a little bit. But the both together really drastically change what is being phosphorylated in the T cells, amplifying the immune response quite substantially. So now I talked a lot about non-small cell lung cancer. What about cancer types where we don't have any immunotherapy responses, much similar to GBM? I'm slowly going to walk my way to GBM, trust me. The next cancer type that we got really excited about was ovarian cancer for many, many reasons, but one of them was it's a very dismal disease. Immunotherapy is not working, very similar to GBM. And it's mostly not working because we don't understand how the immune system really is built normally to fight peritoneal tumors. So what we're doing here is we use a cell line that was originally derived in Bob Weinberg's lab which we call CPAC, and it's using the mutations or over-amplifications of cyclin E1, P53, AKT2, as well as KRAS, and it models an homologous recombination proficient high-grade serous ovarian cancer. And this is one of the most dismal diagnoses you can have if you have ovarian cancer, because not even the PARP inhibitors actually do anything to your survival benefit. The only thing that does work is frontline chemotherapy, but 4 out of 5 women actually recur within 3 to 4 years. So first, coming in with the same premise, can we build a differential model system to understand what is not working about the immune response to ovarian cancer in the peritoneum? We first tested, are these cancers really immunotherapy resistant? Fiona, the graduate student working on this, put the tumors into the peritoneum, treated with dual checkpoint blockade immunotherapy, and this is the survival curve. You can see immunotherapy doesn't work. When she does the same thing and puts the tumors in the flank now and treats with immunotherapy, there's 2 things to note here. The tumors grow, and then even without therapy, there's a period of where the tumor is actually being somewhat controlled. And when we add immunotherapy on top, we really see this reduction in tumor control phase and then slow escape, and we think this is actually an antigen loss phenomena that we see here. So yes, we again build a model system where the flank tumors are sensitive to immunotherapy and the orthotopic tumors are not. And this is ongoing research. This is just a flavor of what we're doing here. Here we then looked at the T cell responses, and we find again that there's more T cells in the IP tumor lesions, fewer in the sub-Q tumor lesions. But the ones in the IP tumor lesions have more markers of activation, but less markers of function. This is very reminiscent of what we've seen in the lung tumor setting. And we still have to do more work on understanding what the T cell phenotype really is. But then Fiona also asked the question, what about the dendritic cells that sit in those tumor lesions? Because as I told you, very, very beginning of the talk, dendritic cells are important to re-stimulate T cells as they enter the tumor. And what Fiona observed was there's an accumulation of a very unique dendritic cell subset, and without going too much into the specifics of dendritic cell biology, typically we would expect dendritic cells up here in this flow cluster and down here in this one, and we would not expect this double positive population, with one exception, the mesenteric lymph node and the lamia propria, because these double positive dendritic cells have been reported to induce tolerance to food antigens in the gut. Yet in these ovarian cancers, what we find is that over time we see an accumulation of these dendritic cells, and our working hypothesis now is these dendritic cells might induce tolerance in the tumor to tumor-derived antigens, and that's what we're pursuing. And we think here in the ovarian cancer space, we might actually have a dual hit where the cancer evades somehow productive priming, and then also whatever is being produced is being further shut down in the tumor microenvironment. Now to GBM. Can we also use the same principle to understand why immune responses against glioblastoma are not working, and what can we do to then make them work? So Amy used a cell line C22A. She engineered them to express the ZS green fluorophore, so we can track dendritic cell biology, as well as synfecal ova antigen, and then treated them with dual checkpoint blockade immunotherapy. And this here is the flank tumor setting. So you can see when we put the tumors into the flank, by itself, without therapy, these tumors are somewhat controlled. But when we layer on therapy, we get 100% cure rates. But again, this is flank. But it gives us the lever that we have a productive immune response against the GBM cell line. When we put these exact same cells, exact same cell number, intracranially into the brain, immunotherapy doesn't work. So we again have a model system where we can really understand what works against a subcutaneous flank tumor and what is missing in the cancer immunity cycle for in the brain. And again, some early indications that we have is that something very similar might be true as we see in the ovarian cancer space, where priming might already be very dysfunctional, But then also in the tumor microenvironment, the antigen-presenting cells, that the T cells see, might also be dysfunctional. And to follow up on Natalie's talk, that merits really a multi-layer therapeutic approach where we enhance systemic priming, and then we layer on a remodeling of the tumor microenvironment. And with that, I would like to thank the people that have done the work. This was really a team effort. Brendan started this. Maria followed up. This is all the lung cancer team. Fiona is working on the ovarian cancer together with Brett and Grace. And Amy and Heidi are our GBM star team. And with that, I would like to take questions.
Thank you, Stefani, for this beautiful talk. Any question? Francisca.
Oh, I was just going to ask, can we speculate a little bit evolutionarily? Why is the brain such a cold place? Is it just because it's so dangerous if something gets up there, and therefore everything that could possibly be, right?
Yeah, I think the brain, so the three organs that we're looking at so far, I think, are cold for different reasons. I think the brain, you cannot allow any swelling or any inflammation because we need the brain to live. So we need to protect it, and only if we really have an insult that is high enough do we get inflammation in the brain. The lung is a single layer epithelium, so if you get too much cytotoxicity, you're going to kill. So in this case, I think it's more the immune threshold to trigger is high. When it triggers, but then the second layer immune suppression is also really high. And that's what we have to combat in lung cancer. And I think the peritoneum is generally super prone for inducing tolerance. And I think we're going to see very different mechanisms at play for all three.
And so then isn't the way to solve this problem then to look what the epigenetic differences are that regulate the coldness of these different must be epigenetic, right? And then try to see whether you could re-engineer whatever that epigenetic
I'm not sure it's going to be epigenetics. I think it's most likely going to be cytokine milieus that determine thresholds for immune activation, and then also how many tissue-specific regulatory T cells are around that can suppress. And this is very cancer-specific, I think. Infections don't typically have self-antigens that they bring along. Cancers have plenty of self-antigens that they bring along. So you will always have DCs that have self-antigens plus antigen CD8 T cells can see. So the Tregs are just right there to say, no, 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 this is something we don't want to kill.
You talked about the brain being cold, and I think the opposite of cold brain is a hot brain, which is multiple sclerosis. You know, demyelinating disorder, multiple sclerosis, we just have lots of inflammation. And I think that just tells you why you have to keep the brain cold, because that's just as devastating a disease as
encephalitis is the other example. Any swelling, any killing of neurons in the brain will have devastating effects.
Thanks, great talk. I was wondering what the antigen presentation networks look like inside the brain. You know, is it different types of dendritic cells, and how do T cells circulate in the brain environment? I just don't know as much about it.
Very good question. We're learning as we go. The first flow plots I've seen, I'm like, Amy, are you sure this is how the dendritic cell populations look? By now we've done enough. There's an incredible skewing towards DC2s in the brain, even just naive brains. This might actually be amplified with a tumor there. What we're missing for the brain is an actual infection model or an MS model where we see how, in a hot brain, does the ratio of dendritic cells really change? The draining part is another really interesting one, and Amy is working really hard with Heidi to find out where does priming really happen, because that's still not fully clear.
Stephanie, we have a very long question from the audience here. Can you read? Because
you're the chair.
It's too long to read, right?
The Treg density. Okay, the question is about Treg density in the mediastinal lymph node. The Treg density was the same, but the proximity of the regulatory T cells to the DC1s was increased in the mediastinal lymph node versus the regulatory T cells were further away from the cross-presenting dendritic cells in the inguinal lymph node. So we think it's a proximity and actual cell-cell interaction feed.
And Stefan, I have a question actually. Yeah. I was wondering if in the lung cancer model, now instead, rather than implanting orthotopically a lung cancer tumor, right, in the lung, you would implant a different type of tumor, let's say a breast cancer, right, there, or anything.
Yeah.
Would you find the same type of tolerance? In other words, do you think that you are detecting here tolerance because you are using orthotopic model, or is it really an innate state of tolerance of those tissues?
We've done a few studies using melanoma to address that early, and it's not fully fleshed out, but it depends on what you compare it to. So we use B16 to see lung tumors versus flank tumors. The immune response against the melanoma metastases in the lung is weaker compared to the primary subcutaneous melanoma, but it is stronger compared to a primary lung tumor. So it's somewhere in the middle, which I think brings me back to there is a component of antigen-specific suppression.
That's exactly what I was thinking about.
For orthotopic tumors, but then the other interesting experiment that we did is we put a lung tumor in first, and then challenged for the subcutaneous tumor. The original priming of the CD8 T cells is dominant, and we cannot reverse course by putting in a second tumor in a more immunogenic site.
Okay.
So which is the more natural history of
it's a mix of components.
Yeah.
Okay, I guess if, I don't know, there are more questions. If not, thank you, Stefani. We move to